This is a lecture presented by the Canadian Bioinformatics Workshops. This work is presented under Creative Commons license. This lecture is part of the Module 4 called More Depth on Pathway Network Analysis, and the lecture will present the ReactomeFI practical lab. On the white space of the slide, beside the workshop logo, some of other logos are displayed. So the Reactome logo is close to the OICR logo because part of the Reactome is working at OICR in Toronto, and the Cytoscape logo is displayed on the right as the ReactomeFI app that we are presenting today is a Cytoscape app. Our learning objective is to be able to perform pathway network-based data analysis using the Reactome app, and the ReactomeFI network is slightly visible on the slide background. The ReactomeFI app is designed to find pathways and network patterns related to cancer and other types of diseases, and unlike the Reactome database that is available for many species, the app is only covering Homo sapiens as a model organism. So this app has two main components. The first one that is explained in the first paragraph is exploring the Reactome pathway diagrams. We can not only explore the diagrams, but we also can overlay information coming from our gene list on these diagrams and perform pathway enrichment analysis. The second component of the app is doing network-based analysis. So within the ReactomeFI app, we access the Reactome functional interaction network that we call the ReactomeFI network. We will review the ReactomeFI in the next slide, and what we can say is that the app can analyze the network modules, perform functional enrichment, and other functions like overlapping FDA-approved cancer drugs targets onto the networks, and also accessing the cancer gene index. This slide highlights the creation of the Reactome functional interaction network. On the top left, we can see that the Reactome FI is built based on non-curated functional interaction, and other sources of information stored in pathway database are going to be added to this source of information, and using machine learning, we will create a big network of functional interaction that contains this curated functional interaction as well as predicted one. So if we go back to the left and the diagram, then we can see what we mean by functional interaction. So the functional interaction will be the lines between the genes, and especially a line connecting two genes. And it's an interaction in which two proteins are involved in the same reaction. So one of the genes in the functional interaction could be the input of the chemical reactions. One of the genes could be an activator, inhibitor, or they could be components of a same protein complex. So if we go back on the right, then we have the full functional interaction network, so containing all the proteins that have at least one interactor. And nowadays, it's about 13,000 proteins. And what we can do with the ReactomeFI app is that we can query our own gene list and create a network that is basically a subnetwork of the full network that contains only the genes in our gene list. So there are two types of functional interaction. So the predicted functional interactions and the annotated functional interaction. So when we construct the functional interaction, then we use known protein-protein interaction, so known functional interaction, and we add some prior knowledge that we found in pathway database, and all this is trained using a naive Bayes classifier to create new predicted functional interaction. So now the app has two components, and the first component on the left is really to browse the collection of the Reactome pathways for Homo sapiens. So you see here like a pathway diagram, and we have seen this in the lectures. And what the app enables us to us is that we can convert the pathway diagram into a network, and we also can convert back the network into a pathway diagram. But we can not only browse a collection of the Reactome pathway, but we also can upload our gene list and do pathway enrichment analysis using our gene list. And we will see, we can explore the results based on the pathway diagram. And on the right, then we can see the functional interaction network. So what we can do is upload our gene list, and then so we will query our gene list, again, this functional interaction network. And so this is at the bottom, the functional interaction network that we have created using a gene list. So so again, the predicted interaction will be displayed as dashed line, whereas the curated interaction will be displayed as solid lines. So these slides show you the interface of the app. And for the first component, where we browse the collection of the pathway. So on the left of the window, we have the list of the pathways, and then we can select a pathway of interest by clicking on the pathway, and the diagram will be displayed on the main window. But we also can do pathway enrichment analysis by uploading our own gene list, and then we will see which pathway are enriched in our gene list. And then when we click on it, then the genes that are in our gene list will be displayed in purple in the pathway diagram. We also can do the pathway enrichment analysis using a ranked gene list and a GSEA, and in a similar way than the defined gene list, the pathway containing that are enriched in our results will be displayed. Another feature of this pathway diagram is that the ability to overlay drug on the pathway diagram. So we can see these two red nodes, which are two drugs, and the red lines will connect two genes, which are the known targets of these drugs. So as I said previously, we can convert this pathway diagram into a network, which is very useful because then we can play with the node shape and node size and color to display multiple layers of information on the same network. Now, the second component is creating a cluster FI network using a gene list. So on the left, so with the green proteins, this is the functional interaction network that we have created with our gene list. So they are connected by lines, which are the functional interaction predicted or curated. And then we have the possibility to analyze this network using different functions. And in this slide, we have decided to cluster the network. So the clustering is based on the number of interactions between some nodes, proteins of the network. And we can see that the network that is now clustered is colored in distinct colors by the different modules. So for this lab that we are doing today, we are going to upload a simple gene list to create the functional interaction network. But here on the right and at the bottom, that you can see that we also can upload more complex format with not only the gene list, but additional columns containing some numerical values. Could be also the survival data. It could be some mutations data. So many options are existing, but we for the lab, we mainly look at the simple gene list. So this main window of the ReactomeFI app display a FI network that we have created with our gene list. And so each green node is a protein, and the black dashed line is a predicted interaction, and the solid line is curated interaction. And then when what you have to do is to right click on a blank space of the screen, and it will display this pop-up window with some options to analyze your network. You can query the FI source to view the source data used to derive the functional interaction. With annotated functional interaction, you can view the source database, experiment type, publication when it's available. With the predicted functional interaction, you will view a table which shows the dataset containing the interaction and an FI score to indicate the level of confidence in the interaction. So this display shows that we also can overlay drugs on the network, and the drugs will be connected to the genes in your gene list by some blue lines. So the drugs here are displayed by diamond-shaped nodes. We can also access the cancer gene index. So in your gene list, it could be that those genes are known to be specific of a particular subtypes, and then you can have this information displayed on the left. And you can do survival analysis, but only if you upload your survival data. We see the diagram here of a Kaplan-Meier curve between high expression or low expression of your gene list and the Cox pH value on the left. And finally, this is an example of the use of this ReactomeFI app using one project where I had some multi-omics data, so omics data from different sources like bulk RNA or ATAC-seq or single-cell RNA. And then I grouped all this gene list into one to do a big ReactomeFI network that I clustered and I labeled using pathway enrichment analysis. We can see the triangle here are the drugs that I overlaid on top of the network. And then I kept the attribute corresponding to the source of the omics data so that I can retrieve if the gene is coming from ATAC data or for example, bulk RNA, and this is an example how you can use this ReactomeFI app in your own project. And this is ending this presentation of the lab.